Welcome to the 8th Annual GIBCO 5 Days of Stem Cells virtual event and today's keynote presentation. It is my honour to introduce our keynote speaker, Dr. Laura Steenpaas. Dr. Steenpaas received her PhD at the Children's Cancer Research Institute Vienna in 2002, researching protein-protein interactions and Ewing's sarcoma. Later, as a postdoc at the Centre for Molecular Medicine, she focused on genomic imprinting at the murine IGF2R Airn locus. Dr. Steenpaas also served as a postdoc at the Department of Pediatric Oncology, Hematology and Immunology, University Hospital, Düsseldorf. Now, as a scientist and PI at the Institute of Human Genetics, University Hospital Essen, she is focusing on modelling of imprinting diseases and retinoblastoma with human iPSCs. Furthermore, Laura is head of the Department of Human and Animal Cell Cultures at the Leibniz Institute German Collection of Microorganisms and Cell Cultures, and she is Professor for Cell Biology at the Technical University Braunschweig, Germany. Please join me with a warm welcome for Dr. Laura Steenpaas.
Yeah, thank you for this kind introduction and for the possibility to present our work here today to this audience. It's the first time that I'm taking part in such a broadcast webinar, and I hope everything will work fine, and I'm sorry for the delay. I will talk about today about the development of a human stem cell-based model for retinoblastoma, which we've been working on in the last few years. And I have to say that all the work has been done in Essen at the Institute of Human Genetics at the University Hospital there, but I've translocated to Braunschweig in May this year. Okay, so the topic of today's talk is retinoblastoma, which is a childhood cancer of the eye. It's a tumor that develops in the retina, and often it's recognized first by the parents when they take flash photographs of their children, and when they see that one of the pupils appear white instead of the typical red eye, which you're familiar with from flash photographs. And the white you see directly through the pupil is in fact the retinoblastoma tumor, which can be spotted like this. Retinoblastoma is a tumor of the young, so it's usually diagnosed under the age of 5. And as I already said, it's often first realized by the parents due to the leukocoria. So somehow I have problems with proceeding my slides. Okay, here's the next one. So an app was developed that enables parents to analyze flash photographs if white reflections in the eye would be just a reflection or could be indicative of a tumor, so that they go to the pediatrician and have a closer look to the eyes of their children. I don't know, this is somehow not working. Retinoblastoma is a rare cancer with an incidence of about 1 in 20,000 live births. And we see about 8,000 new cases per year worldwide, causing also 3,000 deaths per year worldwide, which means about 1/3 of retinoblastoma disease is fatal. The mortality is quite different depending on the country you look at. So we have a 70% mortality rate in Africa, for example, and only 5% in Europe. And there's a clear split between developed countries and developing countries. and this is mainly due to a delay in diagnosis and start of therapy. Sorry. The gene that causes retinoblastoma is the RB1 gene, which has been mapped to chromosome 13q14 in 1986. So it was one of the first genes that has been mapped at all in the human genome. It's a tumor suppressor gene, and it has lots of functions in cellular differentiation, in regulating genomic stability, and most prominent in the cell cycle. And this is what is shown here on this slide. In the cell cycle, RB sequesters transcription factor E2F, or several factors of the E2F family. So they are bound to RB protein in G0, so non-cycling cells, or in early G1 phase of the cell cycle. And as cell cycle proceeds, RB1 becomes phosphorylated by cyclin-dependent kinases and is no longer able to bind E2F transcription factors. So they are free to bind to their target genes and switch them on, so cell cycle can proceed and go on. And of course, if you lose RB protein due to mutations and inactivation, E2F transcription factors are always present and are always able to activate the genes which are responsible for proceeding in the cell cycle, which then is directly linked to tumor formation. Retinoblastoma was the example used by Knudsen to develop his famous two-hit hypothesis of cancer, which applies to tumor suppressor genes. So he plotted unilateral versus bilateral cases, age of diagnosis, and saw a clear shift in time. And he hypothesized by that you need two hits to inactivate the RB1 gene. And this is the paradigm connected to tumor suppressor genes. You have to inactivate both the copies in your genome to start tumor formation. And this is also the case in retinoblastoma. And these two hits can either appear in the same cell of the developing retina, which is a rather rare event, and this will result then in sporadic retinoblastoma, which are seen in about 60% of patients, or 60% of patients present with sporadic retinoblastoma. In the other case, it's heritable retinoblastoma, and there the patients already inherit one inactive copy of the RB1 gene from their parents, either via the germ cells or because a mutation occurs very early after fertilization, which means they carry one inactive copy of RB1 in all of their body cells and, of course, also the retina cells, and they need only one hit more in the retina, in a specific retina cells, to reach complete inactivation of RB1 genes. This results then in the appearance of differences in, does it move? In the numbers of tumors and affected eyes. So the sporadic retinoblastoma usually is unilateral disease, which means we have usually one tumor in one eye, and it's diagnosed at about the age of 2 years, whereas the heritable cases of retinoblastoma usually have bilateral disease, so they develop tumors in both eyes, and usually you have more than one tumor in both eyes. And there's also a special form of retinoblastoma, which is called trilateral, which either comes with unilateral or bilateral tumors in the eye, but in addition, a tumor in the brain develops, usually in the pinealis gland, and this is also heritable RB. So it was clear pretty soon that you need inactivation of the RB1 gene for development of a tumor, and of course groups set out to model retinoblastoma in mice to be able to study tumor formation and tumor development. So several knockout mice were generated, and they've been published back to back in the September issue of Nature in 1992. 3 independent groups generated knockouts of RB1 using different mouse ES cells, using different breeding strategies and mouse strains, and they also targeted different exons and inactivated RB1 by that. And the result was always the same in all 3 papers. They could see when they have a homozygous knockout of RB1 in the mouse, the pups die in utero, and they don't die because of cancer, but because of anemia and neural defects. So this also shows that RB1 has a role in hematopoietic development and neural development in early development. If they kept the heterozygotes, so they were born at normal ratios, and they kept them for a longer time, so they usually developed tumors at the age of 8 to 10 months, but these were brain tumors and not tumors of the eye. So this already indicated that the mouse is probably not a good model for human retinoblastoma, because eye development and development in general might proceed different. But of course, the researchers didn't stop here, but they generated additional mouse models for retinoblastoma, also inactivating additional genes like P53, P107, or P130. And when they did so, and also generated models where they could specifically inactivate these genes just in the retina, and by doing so, they could reach, in the end, tumor formation in the eye of mice. And when they inspected these tumors by immunofluorescence staining, they could see clear differences between human and mouse tumors. This is shown on this slide. So in the top row, you see a human RB tumor, which is negative for RB1. And in the lower row, you see a mouse RB tumor, which was explanted from an RB1 minus P130 minus mouse. And the tumors have been stained for markers, either for photoreceptors, CRX and TR-beta2, or for interneurons of the retina, amacrine and horizontal cells with PAC6 and syntaxine. And you clearly see that the human RB is positive for the photoreceptor markers, but the mouse tumor is not, whereas the mouse tumor is positive for the interneuron markers, but the human tumor is not. So this also shows again that even when the mice develop retinoblastoma in their eyes, it seems to be different from the human tumors that we see and look at. So the group of David Kubrinik used human fetal retina explants and generated an RB1 knockdown using lentiviral transduction to directly see what happens if you inactivate in the RB1 in the human retina. And this is just one image from his paper. You see stained in red LM-opsin, which is indicative for cone photoreceptors in the retina, so they form a distinct layer, shown in red here at the scrambled control. And when you knock down RB1 using 2 different shRNAs, you see green cells appearing, and the green stain is in fact Ki67, so a marker for proliferation. This means you have proliferating cells appearing in the cone photoreceptor layer, which is usually made up by postmitotic photoreceptor cells. So this means knockdown of RB1 in these cells results in induction of proliferation and re-entry into the cell cycle. And this led to the hypothesis that the cell of origin for human retinoblastoma would be an arrestin-3 positive cone photoreceptor of the LM type. And to model retinoblastoma more closely and in a more genuine way, today xenografts are used, so that this, which means that you take a tumor biopsy from the patient and transfer this tumor material to the eye of mice, where tumors will develop. And it has been shown that these tumors are very similar in their molecular type and makeup of the human tumors that have been explanted. But of course, you cannot use these xenograft models to study tumor initiation or development of the tumor, because you already start with a fully grown and established tumor material. So there are a lot of models out there for modeling retinoblastoma, but none, up to now, none is really the optimal choice. And because we've been working with stem cells for a while in Essen, and we came across the Nakano paper in 2012, which was published in 2012, where they showed that you can differentiate human embryonic stem cells or iPSC cells towards neuroretina. We decided to start and develop a stem cell-based model for retinoblastoma. So the upper cartoon just shows how the eye develops in a very brief and schematic way. So the eye is part of the brain. You have an evagination of the optic vesicle first. When the optic vesicle touches the surface ectoderm, it starts to invaginate again. And in the end, you have a two-layered structure, which is called the optic cup, with the inner layer being the neuroretina, where retinoblastoma tumors occur, and the outer laying is the retinal pigment epithelium, which is needed to sustain the photoreceptors. And below you see the images from the Nakano paper showing an evagination and a structure then later at day 26, which very closely resembles the optic cup you see up here in the schematic view. And when you stain these structures for markers, for RPE in red, you see it on the outside, and progenitor of the retinas in the inside. So this shows that human ES cells are able to differentiate into neuroretina and optic cup-like structures in vitro. And of course, if you use these ES cells to model neuroretina, you would like to have it as close as possible to the real thing. And then, so the next slide shows you the structure of the retina in more detail in a schematic way. And in an image taken from optical to coherence tomography, which the ophthalmologists do to have a close look at the retina. So the retina is a layered structure consisting of 7 different neural cell types, and these are organized into these layers. So the light is coming from the top in this case. The first layer you see is the ganglion cell layer, which and the extensions of the ganglion cells make up the optic nerve and channel the light signal to the brain. You have an inner layer consisting of the interneurons, which are horizontal amacrine and bipolar cells. They are used to transmit the signal from the photoreceptors to the ganglion cells, and they are here in the nuclei of these interneurons are in this darker gray layer here in the OCT image. The light gray layer on top is the ganglion cell layer. And then you have the layer of the photoreceptors, which consist of rods and cones. Rods are responsible for dim light perception, whereas cones are needed for color vision. And the humans have 3 types of cones responsible for blue, green, and red color vision. And the photoreceptors are organized in this layer here in the OCT. The dark gray mass are the nuclei, And the brighter layers you see here form the outer segments of the photoreceptors. And the RPE, retinal pigment epithelium, would be here beneath the photoreceptors because they, as I said already, they are needed to sustain them. And as I said before, if you use stem cells to model the retina, you would like to see appearance of all the 7 cell types and, of course, also building up the structure in the retina. And this is, in fact, what happens. These are images taken again from the Nakano paper. You see the ganglion cell layer is the inner layer of the organoid, whereas photoreceptors shown here in green, and the nuclei layer of the photoreceptors appearing in blue, so DAPI stained, form on the outside. So you have a layered structure and you have several cell types present in these retinal organoids. So when we saw this, we said, okay, let's try it and generate knockouts of the RB1 gene in human stem cells and then differentiate them into neuroretina and see if we could use this as a model for retinoblastoma. So we have used the H1 and H9 cell lines, human embryonic stem cell lines, and have generated 2 types of RB1 knockouts. Using the CRISPR-Cas9 system. So we either targeted exon 3 with 1 guide RNA, resulting in random indels and frame shifts in the RB1 protein. And the other strategy was to delete complete exon 1, including the promoter of RB1, so to abolish complete expression of RB1 mRNA and then, of course, also RB1 protein in the end. On the next slide, it's just a brief line up how we did it. So as I said, we used the CRISPR-Cas9 system. We used still plasmid-based or plasmid-encoded Cas9 protein and guide RNAs. We used nucleofection using the AMAXA system into the human embryonic stem cells. And directly after nucleofection, we seeded them to 6-well plates to give them time to recover and to reach confluency. And when confluency was reached and they were split ready, we performed low-density seeding on 10-centimeter dishes. So we seeded about 5,000 cells per dish. And then waited for about 7 to 10 days until the clones appeared, and then we isolated them manually by using a pipette and transferred them independently to 96-well plates. And when they grew up again, we did a split freeze, 1 to 2 split freeze step. So one half we froze, and the other half we transferred to a new 96-well plate and isolated in the end genomic DNA for screening. And on the next slide, you see that we did pre-screening or applied pre-screening either by fragment length analysis for the indels generated in exon 3. By fragment length analysis, you just amplify your target exon and then run it on a sequencer. And this method is able to detect 1 base pair differences, so you have a first indication if indels happened in this exon, and you can select your clones. And the pre-screening for the exon 1 deletion we did by PCR because it's a 400 base pair deletion, so it's easy to spot the fragments on the agarose gel if a deletion occurred or not, and if it would be heterozygous or homozygous state. After the pre-screening, we selected and thought and expanded several clones and checked them again for the type of mutation by Sanger sequencing or in the end by next-gen amplicon sequencing, because by this method we were able to assign the mutations to the 2 separate alleles. And I'm showing here the example of the H9 RB1 exon 3 indel cells, which we used for the which we mainly use for our differentiation experiments up to now. So we have chosen 2 clones, the CO7 clone, which has a homozygous RB1 knockout, so both alleles are inactivated, and the G12LS cells, which are heterozygous state, so 1 intact RB1 allele and 1 allele mutant. And we decided to go with these clones because they carry exactly the same 7 base pair deletion in exon 3, resulting in a frame shift in the protein. So the next slide shows the summary of all the cell lines we generated. As I said, we used H1 and H9 ESCs. We have generated both types of RB1 mutations in both lines, and we always selected clones either heterozygous or homozygous for the RB1 mutation. And as I already said, we used mainly the H9 cells with the exon 3 indel mutation for our studies, and I will refer to them in the following slides. Of course, when you use CRISPR-Cas9 or other techniques of genome engineering on your stem cells, you have to verify and characterize them carefully. So the next slide shows the karyotyping, which is a rather crude method to check for genomic integrity, but is still standard. So these are the H9 cells, exon 3 indel, heterozygous and homozygous deletion with a normal karyotype. We then went on to stain for the pluripotency markers, either nuclear markers, OCT4, NANOC, and SOX2, or the surface markers, SSEF4, TRA160, and TRA181 on the next slide. So the nuclear markers we did by immunofluorescence staining, and the surface markers we tested for by FACS, and they were nicely positive. So from this we already concluded that we have pluripotent stem cells, but of course you have to prove the pluripotency by differentiation assays. And on the next slide is shown the teratoma assay for the H1 cells, which is probably still the best model to use, but of course it's an animal experiment, so for the later cell lines or for the following cell lines we switched to tri-lineage differentiation, so directed differentiation using different media. In both cases we see appearance of derivatives of all 3 germ layers, either in the teratoma assay with showing here with the neural rosettes, structures for ectoderm, cartilage type tissue for the mesoderm layer, and these cells here for the endoderm showing a gland-like structure, or with the directed differentiation FACS staining for marker genes, and we have double positive cells for ectoderm, endoderm, and mesoderm in all cases. So we were confident that our cells are okay, and then moved on to set up the differentiation. One thing we did was also whole genome sequencing, which is shown in the next slide, because we wanted to make sure or check for off targets, which is always a concern when you do CRISPR-Cas9. We first started to amplify and sequence the top 10 predicted targets, but then it appeared to us that the top 10 targets are changing depending on the algorithms you are using for designing your guide RNAs. So we thought, okay, how useful is it? And in the end went for the whole genome sequencing of the H9 RB1 exon 3 indel cells, and we sequenced the parental line, the H9 line, and the heterozygous and the homozygous clone. All of them have around 600 genomic variants that are different to the reference genome. Most of them were common in all of them, so they were private variants to the H9 line, but 58 were not shared with the parental line H9 human embryonic stem cells. So they are private to our genome engineered cells, 18 of them are shared between the heterozygous and the homozygous clones, so this might indicate that they appeared early during the experimental manipulation. And one of these 18 mutations is, of course, the RB1 mutation we introduced. And in the end, you see that we have about 20 private variants to the two clones, but none of these 20 variants was found at a predicted off-target site. So these are just single nucleotide variants appearing during cell culture. So we went on and used these cells for the differentiation into neural retina, and the protocol is shown in the next slide. We are using a protocol published in 2015 by Kuwahara et al. Our protocol is slightly modified to the published one, and this is basically the first step here, that we use dual SMART inhibition and Wnt inhibition in the first 6 days to drive the cells into neuroectoderm direction. We start with the normal human embryonic stem cell colonies, disintegrate them into single cells, and then plate 12,000 cells per well in a low adhesion 96 well plate. We do this for the first day in agar-well medium because that gives the best results for us, and afterwards we change to organoid-forming media containing still dual SMART inhibition and Wnt inhibition, and at day 6 starting to add BMP4. This is then stepwise diluted out and leads to induction of the retinal tissue, which appears here as the bright outer rim in these organoid structures. We kept the induction reversal step described in the initial paper. This should lead to the formation of retinal pigment epithelium in the organoids, but I have to say that we rarely see this, but we still stick to it because that's the protocol we've established and we are using. And in the end, you switch to long-term culture medium, which then contains 10% of serum, retinoic acid, and taurine. And we go until day 152 in our retina differentiations. Depicted here are H1 and H9 wild-type cells. They behave the same. They have the same efficiency of organoid formation, and also the appearance of them is pretty similar over time. So we applied it to our genome-engineered cells, and in the next slide you see the comparison there of them. Yeah, so the wild-type cells are on the top, and then you have the heterozygous line and the homozygous line. From day 1 to day 126, you see the bright outer rim appearing. You see lobes, structures appearing over time. Sometimes these organoids also fall apart into several smaller ones, but they are also still nice and have this bright outer rim. And up to day 126, all genotypes appear more or less the same, so we don't see a difference in gross morphology or efficiency of organoid formation. But when we move on up to day 152, we see a clear difference shown on the next slide, which shows that the homozygous RB1 organoids start to disintegrate. So they lose the nice appearance with the smooth outer lines and the bright rim. So this means at the later time points we clearly see a difference between the wild type and the heterozygous lines and the RB1 knockouts. The heterozygotes here is shown an example what it looks like when the bigger organoids presented here for the wild type line fall apart. This doesn't matter because they are all nice, and when we use them for immunofluorescence staining or other analysis, they give a nice result. So we first then tested the H9 line and our differentiation protocol if we could develop all 7 retinal cell types. This is shown on the next slides. We first stained at day 35 for early markers. So we use Rx and VSX2 for retinal progenitors, CRX for immature photoreceptor cells, and BRIN3 for the ganglion cells. And you see already at day 35 that we have a two-layered structure. We have one layer on the outside and one on the inside, which is most prominently seen with the BRIN3 marker for the ganglion cells. They are organized on the inside of the organoid, whereas we have DAPI-stained cells on the outside. In the outer layer, we find the retinal progenitor cells, Rx, VSX2, and CRX positive. When differentiation goes on, we also tested for appearance of the interneurons, shown in the next slide at day 96 and day 152 of differentiation. These are horizontal cells and amacrine cells. The horizontal cells appear in the middle layer of the organoid. They develop over time. They are rather rare cells, so they don't form a consistent broad layer like the ganglion cells at day 35. Amacrine cells are organized at the inside. They also develop over time. Between day 61 and 126, you see a clear increase in the appearance of amacrine cells. And shown on the right are the bipolar cells and the Müller-Glia cells at day 152. These are the two cell types that appear latest. The bipolar cells also arrange in this middle layer, where you can also find the horizontal cells, and the Müller-Glia form a web throughout the organoid. And on the next slide, I'm showing you the photoreceptors, cones and rods. So the first image is day 61 CRX, which is immature photoreceptors organized nicely on the outside of the organoid. And then we stained more specifically for RXR gamma, day 96, which is an early cone marker. And then later on with arrestin 3, this is a marker for the postmitotic maturing cone photoreceptors, and you see them nicely present and organized at day 152 here on the outer rim of the organoid. Rod markers we use are NRL for early rods, and you also see them nicely organized on the outside in the photoreceptor layer. So we were confident that our differentiation protocol works, our cells are fine, and so we moved on to the comparative differentiation with the three genotypes. The short overview is shown in the next slide. With the numbers for the analysis we did, so as I already said, we mainly used the H9 RB1 exon 3 indel cells, clone C7 for the homozygous knockout, G12LS for the heterozygous knockout. We did a lot of differentiations by now, but for the analysis I'm showing you now, we mainly used the results of three independent differentiations, which we did with all three genotypes. We analyzed 3 organoids per genotype for immunofluorescent staining, and we took 4 images per organoid. So this is wrong here. So it's 4 images per organoid, so that we would be able to quantify our results and not just having the impression you get by eye. And we stained 5 time points from early to late, day 35, 61, 96, 126, and 152 as the latest. And yeah, in the next few slides, I'm guiding you through our results. The first is ganglion cells at day 35. The arrangement is always the same. You have the parental wild-type cells on the left side, the heterozygous knockout in the middle, and the homozygous RB1 knockout cells and retinal organoids on the right side. When staining for ganglion cells using BRIN3, we don't see a real difference. We have the cells nicely present in the inner layer of the organoid, like we showed it before. So there is no difference in the 3 genotypes. On the next slide, we have the interneurons, horizontal and amacrine cells. Thank you. Horizontal cells stained with PROX1, amacrine cells with AP2 alpha, and the time point is day 126. You see both cell types appearing and present in the wild type cells and in the heterozygous knockout organoids, but you see only few horizontal cells, or at least it appears in this image that there might be less in the homozygous knockout, and the amacrine cells are mainly absent in the homozygous knockout cells, so which means RB1 has an influence on retinal differentiation in the organoids. And of course, it's the question if these cells don't develop or if they would develop and die later. And which is also visible here in these images is that we lose the structure in the homozygous RB1 knockout organoids. So with the other images, when you look at the DAPI stain, you also see the main three layers, the photoreceptor on the outside, then an inner layer, and then the layer where the amacrine cells are located in this image. And you don't see this organization in the homozygous knockout organoids. The next slide shows the photoreceptors. Rod photoreceptors stained by NRL on the upper images, so you see them nicely present in the outer layer for the wild type cells and the heterozygous knockouts, but they are a lot less present in the homozygous knockout, and as I said before, structure is lost, so they're not as nicely organized. Cones are present in all three genotypes. They locate to the outside, also in the homozygous knockout cells. So this doesn't look that much difference between the three genotypes. And the next slide shows staining for rhodopsin, which is a marker for mature rods. And you clearly see that we don't have that many mature rods as immature rods stained by NRL in the images before. But in the wild type organoids and in the heterozygous knockouts, you see them present in the outer layer, but you don't see any rhodopsin positive cells in the homozygous knockouts, which means that although we have a few immature rods, they don't mature, and mature rods are not present in the homozygous knockout organoids. We also stained for cones. For bipolar and the Müller glia are still missing, so they are on the next slide. Bipolar stained with PKC alpha and the Müller glia with Vimentin at day 152, because as I already said, they appear latest. You see the bipolar cells in the wild type cells and in the heterozygous knockout. This is all nice here in the middle layer. But they seem to be absent in the homozygous knockout, so they are lost or not formed. Whereas Müller glia looks similar in all three genotypes, so there's no difference here. We did another staining for the bipolar cells, or a double staining, because it's sometimes difficult to identify them and to be sure that this is really a bipolar cell we are looking at. So on the next slide, you see the double staining for PKC alpha and CHEX10. And when you look closely, you see some double positive cells like this one here in the wild type cells and in the heterozygous knockout organoids, but they are completely absent in the homozygous knockout. So we are pretty sure that the bipolar cells are not present. So when I summarize these results from the immunofluorescence stainings, on the next slide, we see or we realize that we lose the structure in the homozygous knockout organoids. They are not organized in the nice layers. Ganglion cells, cones, and Müller glia cells seem to be comparable in amount and localization in all three genotypes, whereas we observe less horizontal cells, amacrine cells, rods, and bipolar cells in the RB1 knockout organoids. And because we didn't want to rely on the results by just looking at the images, we set out to quantify it, to quantify these results by taking the 4 images and generating a macro in image J and running it and counting positively stained cells. And on the next slides, you see these quantifications. We start with the interneurons, horizontal and amacrine. So with the horizontals, it's always the light gray bar is the wild type, the heterozygotes are in the middle, and the homozygous knockouts are on the right side in the dark gray bar. There's no significant difference at day 126 for the horizontal cells. So this is all in the normal range. We see a clear decrease in the presence of amacrine cells in the homozygous knockouts. So this is really true, what we saw in our immunofluorescent images. The next slide shows cones and rods. So it's arrestin 3, in this case the markers. So we see in fact that we have more arrestin positive cells in the knockouts. So we have more cones, but we have less rods also, as we observed in the images, but it's not statistically significant between the wild type and the knockouts. So there might be some sort of variance in these data. But we clearly see that we have more cones, which would fit to the theory that it's a cone cell of origin for retinoblastoma, and when we would assume that our homozygous knockouts are a model for retinoblastoma, we would expect to see more cones, photoreceptors, to be present. We next stained for Ki67 to check about proliferation. If we would really talk about the tumor model, we would expect to see more proliferating cells in our homozygous knockouts, and this is in fact what you see on the next slide. We have the images on the right side, not moving. At day 152, stained in red for Ki67, you see that you have only rarely proliferating cells at this late time point in the wild type and in the heterozygotes, but we have quite an amount of proliferating cells still present, and this is also what is shown in the quantification here with the bars. So this means if you don't have RB1, your cells don't reach the state of terminal differentiation, but they continue to proliferate. Of course, we wanted to know which cells are proliferating, so we did a few double stainings. The first one on the next slide is the cone precursors, so a double staining for Ki67 and RXR gamma. You see the images showing a nice separation between the green and the red signals in the wild type and in the heterozygotes, and quite an amount of double positives in the homozygous knockout organoids. And this is also seen here when you look at the bars in the quantification. So this is highly significant that we have proliferating cone precursors in our RB1 knockout organoids, which also fits to the hypothesis that it might be cone precursors that are the cells of origin for retinoblastoma. We also stained for arrestin 3, so for the more mature cones that are usually post-mitotic. And on the next slide, you see that also then we have some double positive cells in the knockout organoids. So they are positive for arrestin 3, which is a marker for a post-mitotic cone photoreceptor, but we have quite some cells that are double positive, which is also shown here in the quantification. This is a clear difference between the wild type and the heterozygotes, and they appear already early at day 96, these double positive cells, and the ratio doesn't change over time. The last things we checked are the cone subtypes. So the one is S-opsin for the S cone type, stained in green, Ki67 again in red. We haven't quantified these images, but by just looking at them, you see a clear difference. Again, we have a lot more proliferating cells in the homozygous knockouts and almost no green cells, so S-opsin cones seem to be not present. But in the next slide, you see the LM type of cones, which are clearly present in the homozygous knockout cells, and they also stain positive for Ki67. So this means we have proliferating LM type cone cells in our homozygous RB1 knockout organoids, which are not visible in the wild type or in the heterozygotes. So this leads to my final summary and the conclusion. So you see here side by side features of RB tumors and features of our RB1 homozygous knockout organoids. RB1 is a soft tumor, so it's not a solid tumor mass residing in the eye, which might fit, but this is just a hypothesis, to the loose structure we see in the organoids, so that we don't have the nice layers. They disintegrate at the later time points, and they don't form these clear layers like the wild type cells or the heterozygotes. The RB tumors are positive for photoreceptor markers, as I showed you in the introduction, and they show an absence of interneuron markers. And this is also what we see in our organoids. We have a loss of amacrine and bipolar cells, which are interneurons, and we have a gain of cone photoreceptors, especially of the LM type. But we see a loss of rod photoreceptors, and this has been described for also in mouse models. And as I already said, it was hypothesized by the Kubrinik lab that it's LM cones that are especially susceptible to RB1 loss, and that's also what we see in our organoids, that we have a gain in proliferating LM cones. So we are quite confident that our retinoblastoma model in stem cells is a good model and shows essential features of at least initial early retinoblastoma. And this would be then the first human model that would be really applicable to study early events in retinoblastoma formation and in the transition from retinal cells to tumor cells. And the last slide is my acknowledgement slides. So as I already said, all the work has been done in the Institute of Human Genetics in Essen, where I've been up to May this year. And here Denise and Julia have been very active in doing the differentiation and generating the knockout cells. Also Leonie, she's the one who generated the H9 RB1 exon 3 indel cells. And Hannah and Morgan did a great job in doing all the stainings and the microscopy work. And I also want to mention the physicians, Dietmar Lohmann and Petra Ketteler, who provide us with the clinician's view on retinoblastoma and were always very supportive for our experimental project. And so thanks to them, and also thanks to you for listening. And I'm happy to take questions.
Thank you so much for that very interesting presentation, Laura. Such informative work. So throughout the presentation, we have received some questions. We have a few minutes left, so if it's okay, I'll ask you some questions.
Yep, sure.
Thank you. Can you speculate about why loss of tumor suppressor RB leads to loss of some cell types?
Why this is, you mean? So we haven't looked into this in great detail yet. As I said, I would assume that they don't develop, so that there is a difference in the differentiation program if RB1 is present or not. We have started RNA-seq experiments with the organoids, but we are not yet finished with the analysis, and I hope we get some clues out of this. But yes, I think in the end it will be a difference in the differentiation program depending if you have RB present or not.
That's great, thank you. Are you thinking about doing a conditional knockout in ARR3+ cone photoreceptors to isolate those effects from other cell types?
Yeah, that's a good question. We haven't thought about it yet. Yeah, one could think about it to do. Yeah. So as I said, we haven't thought about it yet. I'm not sure about the technical point of view, but the photoreceptors are on the outside, so it would be reachable if you add because I assume that you have to add the inducer then to the media. So it has to get access to the cells, but the photoreceptors are on the outside, so this should work. Yeah, maybe this would be a good idea to look at isolated cell types and couple the knockout to some, but...
Okay, thank you.
I have to think about this a bit more.
Has anyone created a mouse model with conditional RB knockout in photoreceptors? And if so, how does that phenotype compare to the model here?
Yes, so there are several mouse models out there with conditional knockout, but as I said before, it's not just an RB1 knockout, it's always plus an additional knockout of either P53, P107 or P130. So genetic conditions is clearly different. And when they do this, they see development of retinoblastoma, but they also see their own, I think. Yeah, but I think this is what I showed you in the beginning, that these tumors are different from the human tumors, so that they show more presence. So it looks like they would... no, sorry, I think I'm mixing it up. So as far as I remember, there's no conditional knockout in mature photoreceptors, but I might be wrong there. And if you do it in the progenitors, you see appearance of tumors, but they also will switch then. They will have a different makeup of expression than the human tumors.
Okay, thank you. We have time for probably one more question. Are you surprised that the RB ESCs show normal EB formation with derivatives of all three germ layers and no apparent over-proliferation of any cell types or lineages?
Yeah, we were surprised because we expected that the undifferentiated ESLs would proliferate more because they lose RB1. But I think that RB1 has a different function and probably a different role in the stem cells, or that the stem cells have a tighter control of cell cycle, which is not just dependent on RB1. So that this has to be separated, and that the stem cells apparently, at least on the rather broad level, we looked at them, don't care if they carry RB1 or not.
Super, thank you. Well, we're up for time, so I just want to say again, thank you so much for your very interesting presentation, and thank you to the audience for listening, and we hope you enjoy the rest of the event. Have a good day.
Yeah, thank you.